So, as Margaret says, I want to talk about efficacy and safety of new medicines, a human focus. I don't think there's any debate about the fact that there are problems in the pharmaceutical industry at the moment in bringing new drugs to market. There are drugs coming through, and very commonly they seem to suffer ills once they get onto the market. Things happen that were not anticipated. And it is causing a problem with all the major and the minor pharmaceutical companies. The question is why. Drug companies used to be able to produce drugs, get on the market, make huge profits out of them and cure or at least treat many diseases. This appears not to be quite so easy nowadays. What is it that is going wrong? It's a difficult question to answer. It's multifactorial, but one particular aspect that I want to focus on is an over-reliance on animal models. And it's something that I have felt strongly about for many years. I worked at Glaxo for 30 years and did an awful lot of animal experiments in that time. And it became clear to me that you could get almost any answer you wanted by careful selection of the animal in which you use for the test method, and which didn't really seem to me terribly useful. And so I've always been quite keen on using the human biology, the most relevant biology. Now these are our trusted model species, our surrogate species that we use, are used very widely and are required frankly, for regulatory purposes: the rat, the dog, and the monkey. Here is just a snapshot. It's from a paper by Grass and Cinco in 2002. They took a load of data on bioavailability, and they looked at the correlation between bioavailability in those three species and that in man. And as you see, that in man is on the, if I can hold this the right way around. Here we have the ordinate here, human bioavailability, animal bioavailability on the x-axis there. And if there was a good correlation between the species, you'd expect all of those. This is my line here, this dotted line. I did that myself. I would expect all those points to fall on that line. So there's 100% correlation. Now, that is never going to happen. And just the key here, the dogs are the red triangles, the rodents, green circles, primates are the blue squares. What you see is a real scattergram. There is really no tendency to follow that line at all for any of the species. And even with primates, you've got drugs here, this thing down here, which has zero bioavailability in man, has got a high level of bioavailability, this is oral bioavailability, in monkey. Whereas this one here, the reverse is true, and that one. So even closely related species cannot be relied upon. And if you can't rely on them for bioavailability, of course, then you've got big problems when you're looking for further effects, such as safety, toxicity. And can we be surprised when we look at the tolerability of to these species, the rat and the dog, for things that we just take for granted, we eat every day? These things are poisonous to rats. These things are poisonous to dogs. If these foodstuffs were new onto the market and they had to be subjected to the regulatory stringency that is applied to drugs, we would not be able to get chocolate, licorice, Brussels sprouts, macadamia nuts, etc. I mean, these are substances that are poorly tolerated in those species, but in man, of course, perfectly well tolerated and even enjoyed. And primates, frankly, are not much better. And I just want to mention this study in passing, the catastrophic Northwick Park study, which ended so terribly for those 6 individuals. TGN 1412 was subjected to routine testing. It was subjected to all those tests that are required for preclinical safety evaluation. And that included studies in non-human primates at doses 500 times those which eventually proved very nearly fatal in those human volunteers. So again, human, non-human primates, just because we're closely related, does not mean to say that you can rely on what they tell us. So if we can't rely on animals, what can we rely on? Well, the focus of this meeting is on human biology. So what about using actual humans? Nice idea, but how do we go about it? Very broadly, there are 3 ways of tackling the question. You can test compounds in vivo, in silico, and in vitro. Now, in vivo, at the moment, of course, there's a whole swathe of tests that have to be performed before you can get a drug into man. But there is some exploratory work being conducted with microdosing, here using doses something like 100-fold lower than the minimum expected effective dose. And here you can get an idea of the way that the body is likely to handle that compound. It's not 100% secure, but nevertheless you get an idea, using a dose which is not going to be threatening as far as the individual is concerned, get an idea of its metabolic fate, and also potentially some idea of whether it's likely to cause or to produce any toxic metabolites and whether side effects are likely to occur. This is, I think, the evidence in support of this is growing, and I truly believe that this has now got sufficient head of steam that it will form a part of regulatory submissions in the future. I think the future of this, hopefully, is secure. In silico, likewise, there are many computational programs based on human data which are used to predict likely toxicity, side effect issues, and even efficacy. And the more human data that is put into these programs, the more they develop, the more powerful they will be. And they are already proving useful. There are very many commercially on the market that are available and are widely used. And again, I think these now have built up sufficient head of steam that they will be used and they will be more important in the overall testing paradigm. But what I want to talk about is in vitro testing, and particularly the functional side of it. But before I do this, I will mention that very shortly after the incident at Northwick Park, an in vitro test was developed which would have been capable of predicting, the side effects that were encountered in that Northwick Park trial. Had this test been used instead of testing it in monkeys, the outcome would have been very different. At this point, I just want to mention Safer Medicines Trust, the guys here who've initiated this meeting today, who have an initiative to establish utility of human-based test methods. And here it's a question of taking some compounds that have been subjected to the normal, the conventional testing portfolio, as it were, have got onto the market, have got into patients, and have been found to have problems. And the question really is, if we took those compounds, now we know a lot about them in terms of their preclinical profiles in experimental animals and their clinical profiles in man, could we have done a better job in predicting the outcome of these compounds had we used human-based test methods? And this is being supported by the Safety of Medicines Evaluation Bill, which is currently before Parliament, and this is is also supported by an early day motion 569. So things are happening, things are being discussed, and this, I think, is a very important initiative and something which eventually will happen. I don't know when it's going to happen, but I sincerely hope it's sooner rather than later. But it can never be a regulatory requirement until, one, we're convinced that it works better than what we already have, hence the need for the safety, the Safer Medicines People initiative, and two, supply is sufficient. There's no point in making, however good something is, in making it a regulatory requirement if the access to that tissue is a serious bottleneck. Now we get on to human tissue. If we're going to take using human tissue for evaluating the efficacy, safety of drugs, it has to be a need to have. It has to be a stated requirement. It has to be a regulatory requirement. Now, in the UK at the moment, we're currently limited almost exclusively to two sources of tissue: surgical retrieval and post-mortem. So surgical retrieval, this is for medically related surgery. Tissue, organs are removed for medical reasons, and those can form the basis, with the patient's consent, can form the basis of research tissue. Post-mortem, these are people who have died usually within hospitals, from whom there is consent to remove tissues for research. So these are the primary sources of tissue at this time. And that's all very well. They are extremely useful, and they have supported a colossal amount of work, both in discovery and development, but they're not enough. They are limited, severely limited, in a number of factors. One, range of tissue types, particularly with surgery. Surgery is only carried out for certain disorders. There are only certain tissues that are ever removed. So certain types of tissue are not accessible following routine medical surgery. In quantity, surgery is ever more conservative. There's no point in taking out more tissue than you actually need to, and I think there's a strong drive now to remove the minimum amount of tissue you actually need to. So only a small amount can be made available, and of course it has other purposes. It has diagnostic purposes. It is important for record keeping. So the pathologist only gives out that which he or she feels that they can spare, and sometimes that's nothing. In quality, and certainly this applies to post-mortem material, these are dead individuals. They may have been dead for 12 hours, 24 hours, 36, maybe 48 hours. The changes that have undergone by those tissues, organs and tissues in that time, can sometimes make the tissue pretty useless for its primary purpose. Certainly if you're interested in function, you've had it. In condition, and what I mean by condition is a lot of the post-mortem material comes from geriatrics. They die in extreme old age. So it's hardly representative of the population as a whole. Also, tissue only comes out if it is disordered in some way. Therefore, it's very difficult to get control tissue. And frequency, you get it as frequently as it can be made available. And it's not done to order, and this can be inadequate to support a proper program. So how do we tackle this? Well, what I'd like to say is all of these issues can be tackled if we identify and access another source of tissues. And this is tissues from heart-beating organ donors. Now, these are brainstem dead, but heart-beating donors. People usually with catastrophic head injuries, either road traffic accidents or cerebral infarction, whatever it is that's caused it. So there's a cessation of brain function. The body or the individual is still breathing and is maintained perhaps on a pump to maintain that, and their primary purpose is to provide organs for transplant. Now, if at the same time as those organs are being removed for transplant, other organs and tissues can be removed for research, this can be a fantastic resource. These tissues are of extremely high quality. These individuals are obviously cared for because the organs are being removed for transplant into someone else. So consequently, they're kept under in extremely good condition. And if we could remove, subsequent to removing anything that was required for transplant, and transplant must always come first, subsequent to that, anything else. And I would cite, for example, the GI tract. The whole GI tract could come out. It's very seldom used for transplant. This material could be absolutely invaluable in assessing, for example, drug absorption. At the moment we use dogs and rats and monkeys. We've seen the results you get from those. Or alternatively, people do Cacao cells. They're Cacao cell monolayers and look at the absorption of drugs across these. They're not much better. Here, you can get the area you want. You can look at the stomach, you can look at the duodenum, you can look at the ileum, you can look at the colon. You can look at them separately, individually, and look at the effect of, you can look at drugs being taken up, is it unidirectional, bidirectional, look at metabolism within the wall, etc. There's just so much that can be done. And at the moment, this is very difficult to do. And as a source of this tissue, we need to look to UK transplant, or NHSBT as it now is. Now, I went to the website, and over the year between April and the end and March of this year, there were just over 16 million on the organ donor register, 3,500 transplants performed, still about just over twice that many still waiting for transplant, and then up until early September, further transplants performed, but an increasing number. And year on year, this number waiting for transplant increases. And there's no evidence that is going to change. So I believe that there needs to be a collaboration between the Department of Health, NHSBT, and pharma to work together to establish the infrastructure required to improve access to transplantable tissue and at the same time research tissue. The opportunities for researchers, I think, are obvious. Here, access to an essentially unlimited quantity of an extensive range of these tissues that very often are essentially inaccessible to researchers. Obviously, opportunities for UK Transplant and Department of Health. Pharma will be contributing towards the infrastructure. And opportunities for the public, well, more transplant operations and greater confidence in the safety and efficacy of new medicines. And very rapidly in summary, I say, well, you can read it, but our unswerving reliance and belief in animal testing of new chemical entities for safety and efficacy in humans does not stand up to close scrutiny. It's up to scientists to make sure that they do truly validate their test methods and to adopt those which work and to get rid of those which don't. And I believe it's now time to move forward to more human-focused testing for human medicines with particular emphasis on accessing tissues from heart-beating organ donors. Thank you very much.